Beta-oxidation, also known as fatty acid oxidation, is the process in which fatty acids are broken down into acetyl-CoA molecules that then enter the Krebs cycle to produce ATP. ATP is energy that is needed inside the body. This process occurs when glucose levels are low from a carbohydrate source. Beta-oxidation occurs around the same time as gluconeogenesis. Gluconeogenesis is where non-carbohydrate sources, such as amino acids, glycerol, and lactic acids, are used to produce ATP. Adipocytes are specialized cells that store fat in the form of triglycerides. When energy is needed but glucose is low, an enzyme called lipase cleaves the triglycerides, which form fatty acids and glycerol. The fatty acids are then released into the bloodstream, where they are transported by lipoproteins since they are hydrophobic. They enter the cells that can utilize these fatty acids, such as the liver cells, by the process of diffusion. Glycerol ends up being phosphorylated into glycerol 3-phosphate and then converted into dihydroxyacetone phosphate, which is an intermediate in glycolysis. The first step in beta-oxidation occurs in the cytosol, where a fatty acid is converted into acyl-CoA, an activated fatty acid. The enzyme acyl-CoA synthetase, located in the outer membrane of the mitochondria, catalyzes this reaction where a CoA molecule acts as a nucleophile and attacks the carbon on the fatty acid. This ends up forming a thiol-ester bond between the two molecules, creating acyl-CoA. One ATP molecule is used in this reaction, though it's considered two molecules of ATP, since ATP is converted into AMP plus a pyrophosphate that is then cleaved by water to form two phosphate groups instead of ATP forming ADP. A molecule called carnitine reacts with acyl-CoA, thus removing CoA in the process. This reaction is catalyzed by the enzyme carnitine acyl transferase 1, found on the outer membrane of the mitochondria. This reaction then forms acylcarnitine. The acylcarnitine then enters the mitochondrial matrix with the help of transport protein located on the inner membrane of the mitochondria. This protein is called acylcarnitine translocase. Once inside the mitochondrial matrix, acylcarnitine then reacts with coenzyme A to produce carnitine as well as acyl-CoA. This is catalyzed by the enzyme carnitine acyl transferase 2. Carnitine then gets transported outside of the mitochondria to be used in beta-oxidation. For every one molecule of acylcarnitine that enters into the mitochondrial matrix, one molecule of carnitine is exported outside of the mitochondrial matrix. Acyl-CoA then undergoes 4 reactions to produce 2 products. One of them is acyl-CoA molecule that is 2 carbons shorter than the original carbon chain that was used, and you will receive acetyl-CoA. The cycle repeats in the mitochondria using the newly formed shorter chain acyl-CoA until there are only acetyl-CoA molecules. Note that the last cycle to undergo beta-oxidation from a single fatty acid will be from an acyl-CoA molecule that has only 4 carbons in its chain. This will produce 2 molecules of acetyl-CoA. In the next video, I will go over these 4 reactions in more detail.